Well, thank you so much, everyone, for joining our seminar series. And it's a pleasure to kick off our this fall seminar series with our great speaker, Dr. Leng Han, who is a David Brown Chair Professor in Genomic Medicine, Department of Biostatistics and Health Science. And being part of the Brown Center for Immunotherapy in IU School of Medicine. So it's very lucky that Dr. Han has recently joined IU and become very local to us, which is a fantastic opportunity for a lot of learning for our groups. So Dr. Han obtained his PhD from Chinese Academy of Science. He did his postdoc, very, very successful postdoc in Stanford and MD Anderson. And his lab focuses on a lot of omics, multi-omics studies and utilizing that for, of course, studying many diseases, including cancer and precision oncology. He has been very much involved in a lot of biological contribution to the field in the sense of RNA biology, transcriptions, and multiple publications in various top journals, including Nature Metabolism, Nature Immunology, Nature Communication, Cell Reports, NAR, just name it. He has been cited over 20,000 times and have been author of over 160 papers. So it's a great pleasure to have you in this session, Dr. Han. And without any further ado, let's welcome and initiate our seminar.
Thank you so much for the really nice introduction, and really appreciate for the opportunity to communicate with all the colleagues at Purdue University, since you mentioned that we are very close. So hopefully we can establish some collaborations in the near future. So the title of my talk today is Harnessing Big Data for Precision Oncology. So I guess we can be a little bit more flexible. So if you have any questions, you can interrupt me during the presentation or after, and any way you prefer will be fine. And so I will touch about four sections in my today's talk. So first of all, I will briefly introduce the major data resources utilized in our daily research. And then I will also introduce the three major directions in my lab. The first is identify novel therapeutic targets. The second is develop innovative therapeutic approaches. And the third one is the kind of major direction right now is try to understand the efficacy and the toxicity of the immunotherapy. So first, let me introduce the major data resources we used. So this is one of the review we summarized in last year. Basically, we're using the clinical data, basically perform the meta-analysis, try to extract those clinical information from the clinical trials. And then the second piece, the real-world data, is like including those electronic medical record or health insurance claim data. Try to understand what is the kind of therapeutic approaches or outcomes in those patients. The third one is kind of one of our major focus is multi-omics data. It's kind of all different kind of omics profilings. So each of the data resource, they have their advantage and disadvantages. For example, those clinical data, they are kind of obtained from a controlled clinical settings. So they basically have a very direct information for those clinical treatments. But the disadvantage for this kind of data is that most of the time, the key patient level information is missing from those data, or it's very difficult to get. The real world data, in contrast, they have a relatively very large sample cohort. For example, one of our data resources we use can easily reach like 500,000 patient information in the data resources, which is kind of beyond any clinical trials. However, in the contrast, since the data is huge, and the quality and the completeness of those real-world data can be suboptimal. And finally, the multi-omics data, they definitely can provide the molecular insight for those clinical settings. But in the past several years, those information, those samples probably do not have very complete clinical information. But this scenario has been improved in the recent years, that many of the studies starting to provide both the clinical information and the multi-omics profilings. So one of the largest, I should move this one as well, yeah. So one of the largest data resources we use is the Cancer Genome Atlas. This is really a very data-rich data resources. They include more than 30 tumor types, such as the breast cancer, ovarian cancer, brain tumor. And for each of the tumor type, they have the sample size ranging from like about 200 to 1,000, for example. They have about 1,000 samples in the breast cancer patients. And then for each of the sample, they have all this kind of clinical information, such as the age, stage, grade, overall survival. Although the drug treatment data is kind of not completed for these patients. And more importantly, for our bioinformatician, all these samples, they have the genomic profiling, such as the DNA sequencing for the somatic mutations, the copy number variations, mRNA expression, DNA methylation, microexpression, and protein expression. So these data resources really provide us a great opportunity to perform the integrative analysis to understand or identify the potential biomarkers. Other than the patient samples, we also have the other cancer cell line based resources like the cancer cell line encyclopedia. This data resource, they have several hundred cancer cell lines. They have the omics profiling for all these cell lines, and they have anti-cancer drug screening data for these. The other two kind of parallel data resources, one is the Cancer Therapeutics Response Portal, CTRP, and the other is the Genomics of Drug Sensitivity in Cancer, GDSC. Both of these data resources, they have like several hundred of cancer cell lines, and they also have several hundred anti-cancer drugs. So basically, if you combine all these data resources, you probably have like more than 1,000 cancer cell lines, and have more than 1,000 anti-cancer drugs screening data in all these cancer cell lines with all those kind of omics profilings. And another one is the cancer dependency map. Basically, they use the CRISPR screen, shRNA screen, or siRNA screen to knock out or knock down those individual genes to see whether they have the essential pattern for those cancer cell lines. So this data is also publicly available. And more recently, probably the more advanced technologies has become like a quite standard, like single cell transcriptome, single cell proteomics, or even the spatial transcriptomes and spatial proteomics. Basically, this data can provide more information at the cellular level, or even tell you which cell is at which location on the tumor samples. So it's kind of has been utilized a lot in the tumor and other complex diseases. Thinking about those heterogeneity in the tumor and also other complex diseases. The last piece I want to introduce a little bit is the real-world data. This is one of the real-world data called Optum. It's kind of, at IU, we have the access to this one. So basically you can see that they have the health claims data, including more than 67 million people from all the 50 United States. And they have all this kind of different information, what kind of drugs they use, what kind of other information they have. So again, you can see that the sample size is like 67 billion. It's much huge than any other individual centers that can claim, so can collect it. So this one definitely will provide some useful information for our daily research. So with all this, I just mentioned quite kind of very different type of data, like omics data, like this health insurance claim data. So definitely we need some more advanced algorithms to integrate this all this kind of data. So basically, recently we write another perspective in innovation medicine, try to summarize that we use artificial intelligence that we can integrate this kind of multi-omics or even multi-modality data resources, try to get more information and then try to contribute to the precision medicine or precision oncology. Okay. Then I will try to explain the one of the major direction that we try to identify the novel therapeutic targets. So this is basically a summary. Basically, our lab is kind of focused on the RNA sequencing. As you know, that human genome is very complicated, and then for the RNA or the transcriptome, I guess they are even more complicated. Beyond those protein coding genes, those genome can transcribe like different type of non-coding RNAs, like long non-coding RNAs, pseudogenes, enhanced RNAs, snoRNAs, and circRNAs. Okay. Okay. And then after this non-coding or protein coding genes has been transcribed, there's so-called post-transcription regulation, like alternative splicing, E2I editing, and alternative polyadenation. So in the past several years, focus on all these kind of non-coding RNAs and post-transcription regulation, mainly in the TCGA data resources. We try to identify if those non-coding RNAs or post-transcription regulation, they can contribute to the tumor progression or drug resistance, something like that. And we try to identify the targets that we can use them as a potential biomarker or therapeutic targets. Today I just want to focus on introduce one because the time is limited. And this one is enhanced RNA. I think also is Dr. Kazman is kind of very interested in this one. So we kind of committed with email based on this project. So enhanced RNA is kind of a transcription. They transcribe from the enhanced regions, and at the beginning, when the people found this one, they saw this is kind of either the transcription noise or it's kind of a transcription dependent effects. They probably have no functional effects in the human genome. But with the time, many of the researchers found that these enhanced RNAs actually can play very important roles in the human body. They can regulate those protein coding genes quite close with the enhanced RNAs, or even very far away from the enhanced RNAs, so they can play a functional activity in both the cis and the trans ways. So with this in our mind, we try to quantify the enhanced RNAs across all the TCGA samples. So basically, they have the RNA sequencing data. Although the TCGA data, they kind of poly-A enriched, but still they can capture some of the enhanced RNA signals in the samples. Basically, here's the summary result for the enhanced RNAs across different cancer types. On the y-axis, is different cancer type. On the x-axis is different enhanced RNAs. And you can see that there's a few of the enhanced RNAs that they are actually highly expressed in most of the cancer types on the left panel. The mouse doesn't work very well. But anyway, on the right panel is those enhanced RNAs kind of only expressed in certain cancer types. Like you can see that there are more than 5,000 enhanced RNAs expressed at this kind of pattern, basically suggesting that enhanced RNA is very, very cancer type specific, suggesting they probably play very important roles in different cancer types. And then we try to understand what is the regulatory roles of those enhanced RNAs in the cancer signaling pathways. In this figure, on the outer layer, what each dot represents one enhanced RNAs in different cancer types. And in the inner layer is a protein coding genes. Those protein coding genes are kind of the key proteins in the cancer signaling pathways. And then we can calculate the associations between the enhanced RNAs and those protein coding genes. And we observed quite strong correlations among many of these pairs. And many of these protein coding genes actually play important roles in the key cancer signaling pathways, like the Hippo signaling pathway, PI3K signaling pathway, Notch signaling pathway. And you can see that there are many of these kind of strong associations between enhanced RNAs and the signaling pathway key proteins. And further, we want to understand what is the association between the enhanced RNAs and the drug response. So in the upper layer is a different enhanced RNAs. In the lower panel is different anti-cancer drugs. So for enhanced RNAs, because they are associated with the downstream genes, so we know we probably can map them to the signaling pathways. And for those drugs, because the drug targets, they are belong to some of the signaling pathways, so we can map in those drugs based on their targets to the signaling pathways. And then when we correlate with enhanced RNA and drug response, we see again quite a strong associations between the enhanced RNAs and drug response. And to our surprise, some of the associations are kind of beyond the signal pathways. For example, the enhanced RNA target are within one signal pathway, but they are associated with some drugs that they can target to other signal pathways, suggesting that this kind of regulation is very complicated. So with all this global pattern, we definitely want to prioritize some of the enhanced RNAs for the downstream experimental validation. So this is one of the cases that we compare the tumor breast cancer samples with the breast normal tissues. And then we identify this one of the enhanced RNAs named NET1E. It is in the upstream of the NET1 gene. And this NET1E is highly upregulated in the breast cancer samples, and high expression of the NET1E will lead to worse overall survival in the TCGA patient samples. And then in collaboration with Dr. Wenbo Li at UT Health, we basically activated the NET1E in the cancer cell line. And we observed that when we activate this gene, the NET1 expression is also upregulated. And with the activation of the NET1E gene here, we also see the much faster cell proliferation for the cancer cell lines. In contrast, if we knock down the NET1E expression, then the cell proliferation will become much slower, suggesting that NET1E, this kind of enhanced RNAs can increase the cell proliferation in cancer cell line. Finally, we developed a comprehensive data portal for the enhanced RNAs. Basically, this is a very user-friendly. Users can go to the website, click each module. They can pick out the one of the enhanced RNA to see whether this enhanced RNA is expressed in different cancer samples or different cancer types. And then we show which enhanced RNA is associated with the clinical outcomes, like the stage, like the survival. And we also show the enhanced RNA's targeted genes. Finally, it's a kind of association between the enhanced RNA and the drug response. So this work has been led by a former outstanding former postdoc, Dr. Zhao Zhang, and now is a PI at Fudan University. So this paper has been published in Nature Communications back to 2019. Next, I want to talk about develop innovative therapeutic approaches. So basically, as I mentioned, there's a lot of omics profiling data, like the TCGA data resources of the GDIC, CTRP, etc., and also the dependency map. With all these kind of data resources, we definitely can do a lot of things, try to develop innovative therapeutic approaches. Again, I want to focus one of them for the hypoxia-targeted therapy here. So hypoxia microenvironment is kind of a famous hallmarks for the cancer. Basically, when the tumor grows bigger, then in the central part of the tumor, the oxygen level will reduce, causing this hypoxia pattern. And you can see that many of the reviews try to summarize that hypoxia play very, very important roles in the tumor genesis or even tumor treatment. Basically, they are associated with all kinds of cancer hallmarks, like a genomic instability, like immune response, like the survival. But you know that nowadays it is quite difficult to estimate the hypoxia status in the patient sample, or say in vivo. So there's some of the signatures, gene expression signatures that have been developed to evaluate this hypoxia status in the patient samples. One of the cases is a 15 gene signature that it's not developed by us, but we found this in the literature. And then we try to validate whether this 50 gene signature can really estimate the hypoxia status in different cancer types. So this is also the public data. Basically, it's a kind of those cell lines either cultured under normal conditions or cultured under hypoxia conditions at a different time point. And you can see in different cell types, we always observe that this 15 gene signature, we calculate the hypoxia score based on this 50 gene signature, and the hypoxia score always goes up under hypoxia conditions. And similar to this, we even collect many more cell lines, and we also see the similar pattern that the hypoxia scores really become much significantly higher in those cell lines cultured under the hypoxia conditions. And finally, we also compared this 50 gene signature with the other two gene signature that are proposed by different studies and calculate the hypoxia score across different cancer types. And again, we can see that the correlation is really high between different signatures defined hypoxia status, suggesting that hypoxia status is quite robust using all these kind of gene signatures. So with this 15 gene signature, then we can calculate the hypoxia score for all the samples in the TCGA samples, and then for each of the sample, each of the cancer type, we can define samples to classify the samples into hypoxia score high samples or hypoxia score low samples. This is a summary across different cancer types. And you can see that in different cancer types, this pattern could be quite different. For example, in the KRRP, there are very few hypoxia score high samples, but in the head and neck, there are much more hypoxia score high samples. So suggesting that different cancer types, they are quite different. And then with this in hand, we can then make a comparison because we want to know what is the molecular alterations between the hypoxia score high samples and hypoxia score low samples. And you can see because the tumor has quite a strong heterogeneity, so we use a propensity score adjustment to try to control all the confounding factors like the gender, like the stage, like the race, like the age, etc. And then we can make a fair comparison between the hypoxia score high samples and hypoxia score low samples. So this is a summary for the multidimensional hypoxia-associated molecular signatures. And on the y-axis is the different cancer types. On the x-axis is the 6 molecular features that are characterized by the TCGA. And you can see that at the molecular level, it is also quite different. For example, in the mRNA, we observed quite a large number of alterations for the mRNA expression. But for the somatic mutation, we observed very limited alterations between the two groups. And on the cancer type level, we also see different cancer types show different patterns. Like, for example, the GBM, the brain tumor, they have very limited alterations between the two groups, while like THY and TGCT, they have a much larger number of difference between the two groups. And then we also try to understand whether the hypoxia status will change the drug response. So this is kind of, we pick up those clinical actionable genes on the y-axis. So those clinical actionable genes are the drug targets for the FDA approved drugs. And on the x-axis is a different cancer type. Basically, if there's a difference between the hypoxia score high group and the hypoxia score low group, for this clinical actionable gene, we'll give a label a dot here. And different shape means different molecular types. Like round ones means the mRNA expression, square one means protein expression, a square triangle means DNA methylation. And you can see that there's a quite large number of difference between those clinical actionable genes between the hypoxia score high samples and hypoxia score low samples. And because these clinical actionable genes are can be linked directly to those anti-cancer drugs on this panel. So basically, we can also calculate the correlation between the drug response and the hypoxia status to see whether hypoxia status has the direct impact on the drug response. And again, you can see that there's some of the drugs that may become more sensitive under the hypoxia conditions labeled in these green ones. And there are many also drugs that can become more resistant under the hypoxia conditions labeled in these magenta ones. Please keep in mind that traditional view always think that hypoxia will cause drug resistance, but 99% of the literature review report that hypoxia is kind of somewhat bad, and then they will cause drug resistance. So we need to overcome the hypoxia status to make the drug more sensitive. However, you can see in our data, of course, it sometimes causes the drug resistance, but in many of the time, it can also make the drug become more sensitive. So basically suggesting that this kind of global view probably can give us a more accurate picture for the global landscaping. And we are thinking at that time, so basically we can take advantage of this kind of vulnerability of the hypoxia status using those drugs which become more sensitive at the hypoxia status. So in this case, we don't need to overcome the hypoxia status, but we can utilize the hypoxia status to make the drug more sensitive. So of course, the review always wants some experimental validation. So this is a collaboration with Dr. Liu Qingyang's group at MD Anderson. So basically, we pick up several drugs that are calculated in our analysis. And these two drugs are will become more resistant under the hypoxia conditions. and we perform this using the cell lines, culture them under the hypoxia conditions and then examine their drug resistant IC50 value, etc. And we can see that these two drugs really become more resistant under hypoxia conditions. In the contrast, these two drugs become more sensitive based on our analysis. And then we can also see that in our experiment, they really become more sensitive under the hypoxia conditions, basically suggesting that the concept to utilize the hypoxia status to improve the drug sensitivity is doable, is feasible. So to summarize for this hypoxia-targeted therapy, we basically classified hypoxia status across TCGA samples based on 15 gene signature. And we identified multi-dimensional hypoxia-associated molecular features. And then we linked the associations between hypoxia status and drug response. The most important conclusion is that we demonstrated the hypoxia-associated vulnerability to anti-cancer drugs. Basically, we can utilize those advantages of the hypoxia status. So this paper has been published in Nature Metabolism in 2019, and we are very lucky that this paper has been highlighted in the cover story, and later on has been selected in the Nature collection series for Nobel Prize in Physiology and Medicine in 2019. And this work has been led by another very, very talented postdoc, Dr. Youqiong Ye, now is a PI at Shanghai Jiaotong University. So the final piece, but not the least. So we are kind of focusing on the efficacy and the toxicity of the immunotherapy in the past several years. Here's also a summary of what we have done in the past. So basically, we try to understand, identify those biomarkers that can predict the efficacy, or try to improve the efficacy in the immunotherapy. On the other hand, since the immunotherapy has been treated more and more in the patients, there's also awareness for those toxicity like immune-related adverse events for the immunotherapy. So we also published a series of papers try to identify the potential biomarker for the RE, or try to understand what's going on for those toxicity. So what is the immunotherapy? I think this is a very nice review, summarize the two major types of immunotherapy here. One is the antibody-based. Basically, they design antibody to target PD-1, PD-L1, or CTLA-4. So this is called antibody-based immunotherapy. Another one is the adoptive cell therapy, including the CAR-T, TCR, T-cell therapy, etc. Basically, they are activating those T cells, try to motivate those T cells to kill the tumors by recognize the new antigen representing the tumor cells. So back to 2018, when we jumped to this field, because we don't do much experiments, we're thinking what we can do for our first project involved in the immunotherapy. And at that time, there's quite a review mentioned that not only for cancer, but for many other diseases, the kind of male and female bias. So basically, male and female patients are quite different regarding to their phenotype or even the treatment. And back in 2021, there's another very nice review summarized those difference between the male and the female patients in the cancer treatment. But you can see that immunotherapy at that time is not that huge. So basically, they are more focused on the surgery, radiotherapy, chemotherapy, etc., or even targeted therapy. So at that time, there's another two papers published almost like back to back. One paper is published in Lancet Oncology. Basically, they collect a lot of clinical trials data and do a meta-analysis, and then they conclude that male patients will benefit more from the immunotherapy. And just a few months later, another paper published in JAMA Oncology. This paper, they use a similar approach. They collect the clinical trials, and then they perform the meta-analysis. But their conclusion is quite different. They say there's no difference, or say there's no significant difference between male and female patients. So this is very confusing to us. So, that they use a similar approaches. They collect a similar data sets, but their conclusion is quite different. So we are thinking, what's going on here? We try to understand who make the right analysis. And here's a summary. We basically re-evaluate all the data sets that they collected. So you can see in the bottom bar is a data set collected in the Lancet Oncology paper. So the green ones, this data set has been included, and these white ones means this data set is not included. And the second bar is the JAMA Oncology paper. They collected different data set, and you can see that they kind of avoid some of the data set, but pick up some other data sets. And then when we take a close look in each of the individual data set, so basically we can still calculate the hazard ratio for each of the data set. And then if this one has a male bias, we mark them in the light blue. If they kind of a female bias, we mark them as pink. And although, of course, each of the individual study, they cannot reach the statistical significance, but they still have some bias, right? Like a slightly higher, slightly lower in male patients like that. And then when you take a close look on these data resources, you can see that the Lancet Oncology, they include almost all the male bias dataset, but they missed some of the female bias dataset. So you can imagine if you pull all these datasets together, then you probably will make a conclusion that a male will have better benefit from the immunotherapy. The JAMA Oncology, in the contrast, they missed some of the male bias dataset, but they included some of the additional female bias dataset. Then you probably can imagine why they have the conclusion that there's no significant difference between the male and the female patients. And one more thing I want to point out is that at that time, I guess most of the clinical trials are either from melanoma or lung cancer. So basically, the majority of the data resources is coming from either melanoma or lung cancer. And then we saw that this meta-analysis probably cannot solve the problem for this kind of argument. But we want to take advantage of the TCGA data resources to see if there's a difference between the male and female patients for those potential biomarkers for immunotherapy. Please keep in mind that TCGA data does not have this immunotherapy treatment. However, we can evaluate those potential biomarkers for immunotherapy. For example, the tumor mutation burden, the neoantigen, the TCR, BCR, checkpoint, etc. Try to indirectly infer whether the patient will have better immunotherapy or not. And to our surprise, different cancer types also show quite different pattern. For example, in the lung cancer, we see quite high this kind of molecular features upregulated in the lung female patients labeled in these red ones. And melanoma patients, they are in the opposite. They kind of have more upregulated in the male patients. And again, as I mentioned, that most of the clinical trials are either clinical data either from the lung cancer or the melanoma. And you can see that the molecular features of these two cancer types are very, very different. So you can imagine that you pull them together may got quite wrong result, or say not a robust result. And then we expand these two other immune features, like the different very important immune cell abundance, like activated CD4 T cells, activated CD8 T cells, all the all kinds of known immune checkpoints. And again, we can see a similar pattern that lung cancer has much upregulated this immune checkpoints in the female patients, while the melanoma have either no significant difference or they have significantly higher molecular features in those male patients. So taken together, again, you can see that lung cancer and melanoma, their immune features are quite different. And our conclusion from this paper is that we may not put different cancer types for the meta-analysis, especially for immunotherapy, because their molecular baselines is quite different. And then we also think that the omics data with the clinical information is really critical for us to understand how to provide a clinical insight or for those clinical management. This paper has also been led by Yu Chong, and it has been published in Nature Communications back to 2020. So in 2020, we also realized that there's a so-called immune-related adverse events, and this is a very nice review summarized at that time. So basically, the patient received the immunotherapy, but the immunotherapy can also attack those normal organs. For example, they can occur in all kinds of organs in the human body. Like this one, if something wrong with the heart, it's called myocarditis. And then if something occurred in the skin, it's called vertigo. So you can see that this kind of RAEs occur everywhere in the human body. And the occurrence rate and death rate is quite different for these REs. For example, the occurrence rate for the myocarditis is about 1 to 2%. It's a very rare event. However, it's kind of very serious event. Basically, the patients with the myocarditis will cause like 50% of the death rate. In the contrast, vertigos, they probably occur at much higher frequency, like 60 to 70%. But they probably will be a good sign that the patients will respond to the immunotherapy. Sometimes it's not very serious. You even don't need to treat those patients with this kind of RE. So you can see that RE they have even much larger heterogeneity than the tumor. So at that time, we think that it is very, very challenging to study this kind of RE, especially for those rare events. For example, imagine if you want to study the myocarditis, you probably need like 100 patients with the myocarditis. But if you want 100 patient samples with myocarditis, you probably need 10,000 patients with the immunotherapy because the occurrence rate is about 1 to 2%. And on the other hand, normally those affected organs, you cannot cut those affected organs to get samples, right? Because it's just some inflammation there. So all these kind of heterogeneity are difficult to get a sample, and the frequent different occurrence rate all cause a very, very big challenge for studying immune related adverse events. So at that time, we saw we kind of think kind of alternative ways to study these. So basically, we try to use the real world data. Basically, in the I think it's a database named FIRS. It's an FDA approved adverse event reporting system. Basically, the patients themselves, the physician scientists, or even the nurse, they can report those adverse events in the database. From this database, we collect more than 50,000 adverse events in more than 18,000 patients across 26 cancer types. And for each cancer type, we can calculate the reporting odds ratio for the RE. Basically, we know what is the frequency for the RE occurrence rate in different cancer types. And you can see that in the lung cancer and melanoma, they have relatively higher odds ratio. But in some other cancer type, like LGG and GBM, they have relatively lower IRA ratio. And of course, there's some potential bias here, and you can see that because lung cancer and melanoma, as I mentioned, most of the clinical trials or even clinical practice are performed in these two cancer types. So they probably will be recorded in more comprehensive way. But again, at that time, this is the data we have. And then we can try to link this odds ratio with those molecular features that can be used to predict the immunotherapy response or toxicity. So this is one of the results that we collected those like CD8 T cells, PD-1, and then we can calculate them at the cancer type level, and then we can use those this molecular level to predict the odds ratio for the RE. And you can see that this is x-axis is the predicted RE odds ratio, y-axis is the real odds ratio from the field data resources. And you can see that the top candidate are the CD8 T cells, PD-1 expression, etc. And then we build a biovariate model, try to combine them one by one to see if any of the two combinations of the two features that can predict the RE occurrence much better. And among all these factors, we found this TCR diversity and the CD8 T cells plays the get the best performance. And the correlation here is about 0.75. And then because we have the TCGA data resources, we always want to expand to other molecular features. So basically, we screen or use the similar approaches to screen everything across the whole TCGA samples. And this is the top 10 candidates like LCP1, like GZMP. And again, we do perform a bivariate model to see if any of the combination of the two genes can predict the best. And very luckily, we found these pairs, LCP1 and ATP-GK, they can even predict much better than the CD8 T cells and the TCR diversity, suggesting this is a really good biomarkers. And we take a close look on these two genes. Actually, they are not kind of random genes. Both of the genes are actually highly involved in the TCR activations which makes a lot of sense since that when the immunotherapy treated the patients, then they can activate the T cells. If there's activation of the T cells, they probably will attack the normal organs, such as to cause the ARE in the patient. And again, of course, the reviewer always want to see some of the experimental validation, at least to see that overexpression of these two genes in the patients with the RE. So basically, we collaborate with our collaborators and then pick up the samples with RE or without RE. We get 14 for both for patients with RE and 14 for patients without RE. And I want to emphasize this is really a tough, really tough work. That our collaborators screened more than 10,000 patients, get like these 14 very good ones for the experiment, and we performed a staining to see LCP1 and ADPGK expression in the tumor samples, and we can see that those patients with RE really have much higher expression for the LCP1 and ADPGK like this, and this is one representative images for the staining result, and this is a summary. And you can see that for those patients with RE, they really have much higher the LCP1 expression than those patients with RE. ADPGK is the same. And then we combine these two together, and we can see also much higher, significantly higher values for LCP1 and ADPGK. To summarize for this one, we actually combine the pharmacovigilance data, also real-world data, and omics data, try to identify the potential biomarkers for the RE, which is very, very difficult to achieve using the traditional ways that collect samples, do the sequencing or something, and identify the potential biomarkers. And then we show a bivariate regression model of LCP1 and ADPGK that can be used to predict the RE occurrence. So this paper has been published also in Nature Communications back to 2020. So with this potential biomarker, we definitely want to ask more questions. For example, that sometimes the cancer patients, they were treated with the antibiotics. So we want to know whether those patients treated with the antibiotics will have a higher chance to get RE or not. So this is one of the in-house patient cohort. So basically we observed that in the lung cancer patient, if they treated with antibiotics, they probably will have higher chance to have the RE. We observe a significant signal here, but we didn't observe the significant signal for some other cancer types. I just want to emphasize that if we don't see signals here, it doesn't mean that there's really no signal here, because the sample size is really small, so we probably cannot see anything here. So again, this still motivates us to continue to collect samples to further evaluate whether there's an association. But in the lung cancer, we definitely observe a strong association between the antibiotics treatment with the RE. And then with the FIELS data, we confirm these observations. Basically, in the lung cancer, we have a much larger sample size. We observe that those patients treated with antibiotics, they will have much higher chance to get the RE. Finally, we try to use the omics data to explain this. So basically, we perform this because we can calculate the microbiome diversity in the TCGA samples, and we can also calculate the associations with those RE-related factors or potential biomarkers. And you can see that there's a very strong negative correlation between the microbiome diversity and those RE-related factors and pathways. So we kind of propose a hypothesis like this. So when the patient has been treated with antibiotics, their microbiome diversity will be reduced. I think this is easy to understand because antibiotics will kill the microbiomes in the human body. And because the microbiome diversity is kind of negatively correlated with the RE-related factors, when the microbiome diversity reduced, then those RE-related factors and pathways will be increased, such that they will increase the RE risk. Of course, this is just a hypothesis. We currently do not have more evidence to prove this, but this is something that we hope that we can investigate in the future. To summarize for this part, we show associations between the antibiotic usage and increased RE risk. We try to propose a hypothesis to explain this that this may be due to the decrease of the microbiome diversity. And we also want to say that administration of the antibiotics should be very carefully evaluated for those cancer patients. But of course, we don't want to say that don't use the antibiotics for the treatment for the patient. But this should be, very, very carefully evaluated. And this part has been published in Journal of Immunotherapy of Cancer in 2022. So I talk about the antibody-based immunotherapy a lot for the toxicity. And we also recently shifted a little bit to the adoptive cell therapy. And you can see this is another very nice summary from the Nature Review Drug Discovery. And you can see in the past several years, the clinical trials for this kind of adoptive cell therapy is increased significantly, especially for the CAR T. So basically, CAR T is they can recognize those antigens presented in the tumor cells, like on the right panel. And you can see if the new antigen presented in the tumor cells, the CAR T cell can recognize these tumor cells and then kill the tumor cell. However, if the normal cells also presenting this new antigen, then CAR T cells can also recognize these normal cells, and then they will also kill these kind of normal cells, which may cause on-target off-tumor toxicity. So back to 2020, a very nice paper published in Nature Biotechnology. Basically, they try to evaluate all those CAR targets to see whether they are highly expressed in the normal tissues. Basically, the idea is that if the normal tissues does not express all these CAR targets, then they will be very safe for the patient to use the CAR T therapy. And here's their result. Basically, they evaluate the GTAX RNA expression, HPA RNA expression, and HPA protein expression. And you can see on the left part are those CAR targets that they are rarely expressed in the normal samples, normal tissues, basically suggesting they should be relatively safe. But on the right panel, those CAR targets are actually expressed at a very high level across different tissues, suggesting they may cause some of the off-target tumor off-target issues that the clinicians need to be aware for this potential toxicity. And in the same year, another paper published in Cell, basically they using the single cell data in their mouse model or even in their human samples. Basically, they show that this kind of CD19 is a well-known CAR target. Basically, they show that some of the CD19 positive populations are occur in the human brains. So although this rate is very low, the proportion is I think it's less than 1%. But they saw this will still cause those neurotoxicity in the brain, and they have a mouse model to try to further confirm this. So with all these two papers together, we're trying to think that we can, this NBT paper is really nice, but they didn't touch at the single cell level. You can see that if the cell population is really small like this way, their method cannot even identify this potential toxicity issues. So we are motivated by these two papers, and then we say we can do some similar approaches and then try to evaluate those CAR targets, whether they are expressed in the normal tissues at the single cell level. And at that time, very likely there were two large-scale single cell dataset has been released. One is from the human cell landscape, one is from the adult human cell atlas. Both of the data resources include different normal tissues and thousands or even 10,000 of single cell data. Basically, we use these two data resources, and then we can evaluate whether those CAR targets are highly expressed in those normal tissues. So here's the result, and you can see that on the x-axis is a different CAR targets. On the y-axis is different normal tissues. Again, on this panel, those genes are kind of lowly expressed in most of the tissues or cell types, suggesting they should be very relatively safe. But on this side, they are actually highly expressed in most of the tissues, and then probably they will cause some problems when treating with the patients. So one of the examples we want to say here is the EGFR. You can see that it has been kind of applied in multiple clinical trials, but you can see that EGFR is highly expressed in many of the cell types and across different normal tissues. So this is one example. Basically, at the background sequencing, you can see that EGFR is highly expressed in both skin and lung, and they can be captured by the single cell data. But this one, EGFR is kind of lowly expressed based on bulk RNA sequencing data in the gastrointestinal. However, using the single cell data, we identify them in both of the data set. Basically suggests that EGFR may cause some problems in the gastrointestinal region. And we also check one of the clinical trials. They use the EGFR CAR-T design, and they really report one of the adverse events in the gastrointestinal events, basically suggesting that really single cell data can help us to monitor this kind of potential adverse events in the CAR-T therapy. Of course, as a bioinformatics group, we also want to develop some of the data resources that can be utilized by anyone else in the world. So basically, we developed this data portal for the expression of the landscape of the CAR targets at the single cell resolution. And the users can go to our website, basically simply click all these modules, and then they can get the expression levels for those CAR targets across different normal tissues. So this paper has been published in Cancer in 2021. So to summarize for this part, we have tried to use the real-world data and omics data to identify the biomarkers for the RAEs. We show associations of the antibiotic treatment with the RAEs in cancer patients receiving immunotherapy, and we'll also show that on target of tumor toxicity of the CAR targets at the single cell level provide a more accurate estimation for the potential toxicity. Most of the work has been led by outstanding former postdoc, Dr. Yin Jin, and now he's a PI at the Guangzhou Institute for Health. And two of the current postdocs, Jingwen Yang and Yuan Liu, also help a lot in the projects. Finally, I would like to take this opportunity to acknowledge for my collaborators. And you can see this is a long list, and I should say this is just a partial list. I had a great fortune to work with a lot of great investigators to discuss the functional characterization, biological insight, and clinical impact for all our projects. And also my great team. This photo has been taken back to, I think, 2019, before the pandemic at UT Health. Most of the members here are already, found their effective positions right now. And this is the current group members that are working on the exciting projects right now. And we also like to thank the funding support from CPRI, NIH, Brown Center for Immunotherapy, and Cancer Center. Thank you so much for your attention, and I would be happy to take any questions.
Thank you so much, Dr. Han. So any question from audience? Of course, I have a lot of questions, but I just want to make sure that first audience get a chance to ask any question. Please feel free to unmute yourself and ask questions if you have any. Maybe I would kick off with one, and then we can go from there. So absolutely great set of works that have impact in patients. So a couple questions. One of them is that, do IRAEs, these adverse events for immunotherapy specifically, do you think that they have association with tumor response, meaning that those who have been having more adverse events have better tumor clearance anyway?
Yes, this is a really nice question. I think yes or no for this to answer this. So there's a lot of clinical trials try to link these adverse events with the response. As I mentioned, some of the papers shows that there's sometimes there's rashes on the skin. They are kind of linking to the better perform response in the patients. But this is still arguable. We actually recently write a review paper in Nature Review Clinical Oncology. We summarize all the studies in the past several years to show the conclusion is quite different. Some studies says there's a positive correlation. Some studies doesn't observe any associations. I guess there's I think once they have the RAE, definitely they should have the activation of the immune system. But whether those activation of the immune system really lead to the better response is hard to say. Yeah. So yeah.
That's fair. Any other question?
Okay. I have one question about the enhancer RNA. So you showed us the link between the enhancer RNAs and drugs. So since I'm working on the 3D structures of molecules, I'm wondering if this connection is a physical interaction or the drug is probably just influencing a downstream of enhancer RNA.
Yeah, for our data, it's just a correlation. So we don't have the data to go that deep to consider those structures. But I guess most of the time is enhancer RNA regulate the protein coding genes and then affect the protein coding gene and associated with the drug. That part of probably will kind of be impacted by the structures. Yeah.
Okay. Yeah. Thank you for the clarification.
Sure. Thank you.
So I have one more question. Anyone has a question?
I think in the chat there's one question.
Yeah,
Maybe I can answer. Yeah.
Yeah, that would be great.
Does TCGA use a non-invasive methods to collect its data? I should say this is invasive. Basically, for those tumor, they cut the tumor out and then they sequence those things. The tumor sample is much easier to get because the patients always want to get rid of the tumor, right? So basically, tumor sample is much easier to get. So basically, this is, I guess it should be invasive. Of course, I think the TCGA also get the blood samples. Those are kind of non-invasive. Because they need to do the genotyping for those kind of things. But for tumor samples, RNA sequencing, exon sequencing, those are invasive methods, cutting the tumor out. Yeah.
Okay. So,
Go on.
so yeah. So they use whole exome sequencing, I presume, for collecting the data, which is how would you compare that with some of the recent advancements in techniques such as liquid biopsy using circulating tumor DNAs and cell-free DNAs? Because that is a field that is rapidly gaining traction. So like, would that help in any way in, identifying tumor burden for ICI, since it's non-invasive and it's relatively cheaper and easier than whole exome sequencing?
Yeah. Okay. To be honest, I'm not touching any of the circulating DNA sequencing yet. So I probably is not a good person to answer this question. But according to my very low level of understanding, so basically, I guess the different samples, they have different purpose. For the circulating, they kind of more like early detection. Basically, if the tumor is not coming that far, you try to use this way to detect whether the patient have the tumor or not, right? But TCGA data is more likely they provided like what's going on within the tumor, and then you find a way to treat it. I guess what I heard is that especially for those metastatic tumors, there's a quite a difficulty to treat those samples, treat those patients. So I guess that now it's kind of shifted a little bit that try to identify the cancer as early as possible. I mean, the early the better. If you identify identify cancer at a very early stage, it's always much easier to treat the patient. I think surgery will be enough, not necessary to involve those chemotherapy, immunotherapy. So I guess that's the idea shift right now. Try to detect the cancer much early using the circulating tumor DNA. But on the other side, I think there's still, because that circulating DNA is still, because the amount of those tumor materials is much smaller than the tumor sample. So there's still this kind of sensitivity or specificity issues. So it's hard to say based on this data. You need to develop a very robust approaches to really characterize those circulating tumor DNAs. Yeah, I guess that's the challenging part. Yeah.
Okay. Thank you.
Sure. Yeah, again, I'm not an expert for this, so I cannot. My answer may not be right. It's just my understanding. Yeah.
So let me ask one other question while we have you. So you did a fantastic job in hypoxia status signature, and showing how it's associated with different properties in cancers. Did you also associate that with survival data and whether or not that hypoxia or hypoxia score would be associated with survival in cancers?
Yes, I think that is. I think we have done this in our paper, and also this is well known that hypoxia will lead to poor outcomes in the patient overall.
Okay.
Yeah.
See.
So that's why everyone is talking about trying to overcome the hypoxia, because the hypoxia tumor really. I guess this is also biased because normally hypoxia tumor is much larger, right? So it's easy to understand that hypoxia patient will have worse outcomes. Yeah.
That's fair. Any other question from audience? So if there's no other question, let's thank Dr. Han for his time and great presentation. And
Thank you so much for the opportunity.
thank you
Really
so
appreciate
much.
it.
Thank you very much.
And hopefully we'll see you in person soon.
Sure, yeah, that would be great. Thank you so much.
All right, thank you
Thank
so
you very
much. Bye, everyone.
Thank you. Have a
Bye.
nice day.
Thanks, bye.
Thank
You
you.
too. Bye.
Thank you.